It's a pleasure to talk at this last session on what's potentially in store in the future for treatments of kidney cancer. So as we've talked about in this meeting, we now have a pretty crowded algorithm with at least 8 different treatment approaches for patients with kidney cancer. But I think it's fair to say that we still have patients who don't respond to therapy, respond for a short period of time, and nearly everybody becomes resistant to therapy. So despite this plethora of treatment options, we still need new therapies and new therapeutic approaches. Here are some of the listed opportunities, many of which are talked about in this meeting, and I'm going to focus on novel targets and treatments in the front line and in the VEGF receptor TKI setting. Dave McDermott already mentioned PD-1 and PD-L1, so I'm going to try to quickly cover the 5 targets in yellow here. So first of all, HIF-2 alpha, as it turns out, is the dominant oncogene in clear cell kidney cancer. And we know this because genetic analyses of many kidney tumors suggest that in addition to the VHL loss in chromosome 3, that up to 40% of tumors actually have lost chromosome 14 where HIF-1 alpha is made. And those tumors that are HIF-2 only appear to be more aggressive in some analyses done by immunohistochemistry. And in fact, in functional studies, HIF-1 alpha appears to behave like a tumor suppressor. By that I mean if you put HIF-1 alpha back into a HIF-2 only tumor, you see decreased proliferation. And if you block HIF-1 alpha in a tumor that has both HIF-1 alpha and HIF-2 alpha, you see increased proliferation. So trying to find something that actually can inhibit HIF-2 alpha has been a major priority of the kidney cancer program at the Dana-Farber Harvard Cancer Center, and we've been working on this with 3 different approaches. One using siRNA targeting HIF-2 alpha, work being done in the SPORE project supported by Bill Kalin. The other is trying to develop small molecule inhibitors of HIF-2 alpha using a chemical screen, and work done by Othan Iliopoulos. And those are coming along, but they're still in preclinical stages. I'm going to focus on the third concept, which is trying to inhibit HIF-2 alpha by inhibiting TORC2. So as it turns out, the rapalogs, everolimus and temsirolimus, that inhibit TOR usually just inhibit TORC1, and as a consequence only inhibit HIF-1 alpha. If you want to inhibit HIF-2 alpha, you need to have active site inhibitors that inhibit not only TORC1 but TORC2, and there are a number of those molecules that are currently being investigated. One is BEZ235, a molecule from Novartis, and this is work done by Dan Cho in a mouse model that suggests that using a mouse tumor xenograft, if you treat with rapamycin, you see some benefit, but if you treat with BEZ235, you see much more pronounced anti-tumor effects. And further, while both rapamycin and BEZ are able to block phospho-S6, indicating they're both hitting their targets, only BEZ235 was able to block HIF-2 alpha. So this type of data has led to 2 trials, one which hopefully will get underway shortly, a phase 2 biomarker trial of BEZ235 in patients with kidney cancer at the Dana-Farber Harvard Cancer Center, and a study of a Genentech molecule that's a dual PI3 kinase active site TORC1, TORC2 inhibitor, GDC0980, versus everolimus in the VEGF receptor TKI resistant patients. This trial is well underway, and hopefully we'll have data to report next year. Another good target to look at are the angiopoietins, and they represent a potential alternative way of driving angiogenesis in the setting of blocking of the VEGF pathway. And a molecule that's been studied that's an angiopoietin blocker is a soluble receptor for the angiopoietins, blocking the Tie2 receptor, binding of angiopoietins to the Tie2 receptor, AMG 386. And angiopoietin 2 can serve as a biomarker, a surrogate marker in patients with kidney cancer treated with VEGF pathway inhibitors. As shown here at baseline, can be elevated, it goes down with response, and also comes up at the time of resistance in the majority of patients. And the question is some of that resistance being driven by angiopoietin 2. So there have been 3 studies that have been conducted, a phase 1 study of AMG 386 plus sorafenib, in which there was a 42% response rate in patients with kidney cancer, which was impressive for sorafenib. Unfortunately, the randomized phase 2 trial that Brian Rinney led, looking at sorafenib plus AMG 386, showed no benefit for the addition of AMG 386. So that certainly soured people on this particular molecule. On the other hand, many people believe that the dose of AMG 386 was too low in that trial, and there's an opportunity to re-look at this with a phase 2 study of sunitinib plus AMG 386 at multiple dose levels that will be presented at ASCO next month. And as a sneak preview, there will be a very impressive response rate and PFS rate in that particular trial, potentially causing rethinking of the role of AMG 386 or angiopoietin inhibitors in this disease. Another thing that we've learned from our animal models is that resistant tumors show an increase in the expression of TGF-beta associated genes, and that's sort of shown here in this busy slide, but this pattern here suggests a predominant increase in things associated with TGF-beta. And that prompted Rupal Bhatt at our institution to look at inhibitors of activin-like kinase receptor. And this is a type 1 TGF-beta receptor superfamily member that's expressed on activated endothelial cells. Its loss genetically leads to the hereditary syndrome, hereditary telangiectasias, or Osler-Weber-Rondeau, and a fusion protein that is a mimic of the receptor binds to and neutralizes the activity of the 2 ligands for the receptor, BMP9 and BMP10, and has been shown to exhibit anti-angiogenic activity. So Rupal looked at this in some of our animal models and was able to show that the combination of ALK-FC plus sunitinib produced better anti-tumor effects in one tumor model, but not too much activity as a single agent. And in the second tumor model, it had activity as a single agent equivalent to sunitinib, and the combination was significantly better than either agent alone, with disease stability out to close to 50 days. And in looking at a perfusion MRI, you could see that the combination of sunitinib and the ALK-FC had superior anti-tumor effects or anti-perfusion effects compared to sunitinib alone, as shown here in this particular graph. So there a phase 1 trial, the ALK-FC, is currently underway. There's no data in kidney cancer patients, but a phase 2 clinical trial is being considered with ALK-FC together with a VEGF receptor TKI. And there's another ALK antibody being developed by Pfizer that is also in clinical investigation. The final target and treatment I wanted to talk about is the MET pathway. And as I mentioned yesterday, sporadic clear cell kidney cancer has dysregulation of the MET pathway. VHL loss results in increase in HGF-driven invasiveness, and multiple shRNAs against MET preferentially inhibited the viability of VHL null kidney cancer cells in a preclinical work by Bill Kalin. So we have this molecule, XL184, or Cabozantnib, which is a combined MET and VEGF pathway inhibitor. And Tony Choueiri led a clinical trial looking at 25 heavily pretreated patients who'd received prior VEGF receptor agents or mTOR inhibitors, some who had bone mets, and saw responses in 7 patients, or 28%, and a very impressive median PFS of around 15 months, and there were responses in bone. Here's the waterfall plot showing a lot of tumor shrinkage with only 2 patients showing resistance up front to this treatment. And here's an example of response of a pleural-based metastasis, as well as a response in bone. So these are 5 targets that are potentially worth considering. In addition, there are other potential targets that are worth exploring, including whatever this is on chromosome 5 that's frequently activated in kidney cancer, as well as whatever is potentially lost in chromosome 9, as well as other things that might be seen on chromosome 3, such as PBRM1. And in conclusion, I think there are multiple new targets that are available for exploration in these patients. Some of these are potentially for initial treatment with or without VEGF receptor TKIs. Others might be more appropriate at time of resistance. And hopefully by establishing new targets and new therapies, we will be able to produce more durable responses and have opportunities for more personalized care. Thank you very much.